Welcome to another lecture. This lecture is about blue light responses and blue light receptors. So far we have talked about responses to white light, as for example in photosynthesis. And we've also talked about red light specific responses, as in the case for phytochromes. There are certain responses that are specific to blue light, and they're not mediated by phytochrome, and they're not mediated by chlorophyll or any of the photosynthetic response pathways. So here's an example of a blue light specific response. This is the phototropic response. So in this example, you see a maize coleoptile, that is a little tiny sheath of tissue that comes out of the ground when, for example, a maize corn, like a corn kernel, germinates. And inside are the leaves, the original leaf, the cotyledon. So in monocots, grasses for example, the first thing that comes up is this little sheath of tissue, and that's called the coleoptile. And that's been used widely for studying phototropic responses or gravitropic responses. So what you see here is a time lapse. So this is just one coleoptile. It looks like, what, 10 or so, but it's just time-lapse and then superimposed. And the light is coming from this side, the blue light is coming from this side. And so, as you can see, the coleoptile over time bends towards the blue light. And just like with other receptors of light, what you can do is you can graph an action spectrum so that you can find out what specific responses are specific to certain wavelengths. And so just like we did this for phytochromes, we can here now look at an action spectrum. And in this case, the action that we are graphing is the curvature, the phototropic curvature per photon relative to 436 nanometers. So that is just to say that 436 nanometers was used, and said, so anything relative to that, so that's set to 1, I guess that's here. And then anything set to 1 is relative in its intensity, in its ability to cause curvature with respect to that particular point. So you can see that in the blue light spectrum, there's a lot of activity that leads to this particular response, phototropic response. And so just like with all action spectrum analysis, to find a candidate gene that initiates the phototropic signal, one would look for a protein with an absorption spectrum that matches this action spectrum. So a receptor, a photoreceptor that absorbs at exactly these types of wavelengths would be the one that you would expect to have in these types of wavelengths a response, in this case the response of phototropism. So the question is of this lecture, what are some blue light responses and what are some receptors for blue light responses? So in order to do that, what we will have to do, we have to do a little side trip here, and we have to look at some ways how to quantify RNA. We haven't talked about that in this class. I want to make sure that everybody's on the same path here, on the same page with respect to understanding molecular analyses, molecular blots, and other types of techniques. So the first type of technique that I want to spend a few minutes on discussing is one that's called northern blotting. So with northern blotting, you can, just like with the other quantification methods that I want to discuss in the next few minutes, you can measure how much RNA there is of a particular type, of a particular gene. So the way that this works is that you grind up the tissue, you isolate RNA from the tissue, then you separate the RNA on a gel, on an agarose gel, just like a PCR gel, and then you blot that. I'll show you a picture in a second what I mean with that, but essentially you put a membrane on top of that gel to suck the RNA onto that membrane so that you don't have to work with this wiggly jiggly gel. And then you identify the particular gene that you're interested in by using a labeled complementary DNA strand. So what is a labeled complementary DNA strand? So let's say we have here our denatured RNA, and that's the same as here. So this is the target RNA with a particular gene sequence. So in order for us to find that, we can make a probe, we can make a piece of RNA or DNA that is complementary to the sequence of our target. And so this would be something that you synthesize either by PCR. You can send away for this to be synthesized by a lab. It's very quick and easy to do. You can do it yourself. You don't need to send it away. And as you're doing that, let's say you were to do this with a PCR reaction, making this probe, you could add a certain amount of labeled nucleotides. So in this case, all the cytosines are radioactively labeled. So they have a little radioactive label on, for example, by having a different weight for the isotope. Or these days, more frequently used than radio labels are fluorescent labels. But regardless of how you do it, you label one of the nucleotides, you incorporate it into the probe. The probe is now complementary to the piece of RNA that you're looking for, so a particular gene. And then when you mix those two together in some way, and I'll show you a few ways how you can mix the target RNA and the probe together, then the binding of the probe to the specific region that it's complementary to will then allow this to stick. You can wash off everything else that's bound non-specifically. And then you can visualize this somehow. So, for example, with radioactivity, you would use a film, and then the film, photographic film, would be blackened by the radioactivity. Or you can use, fluorescence. You can also do it the same way that you have film that is exposed by the fluorescent label, or the fluorescent label might be colored, and you can actually view it with the eye. Okay, so let's take a look at how this northern blotting gets done step by step. You may have heard of southern blotting, and I know you've heard of western blotting, and they all have very similar, sort of a similar setup, but there's different things about them. Southern blotting is for DNA. That was the original blot that was invented, that kind of method, by Edwin Southern, who got the Nobel Prize for that. And then northern and western blots are just sort of, people said, oh, there's a southern blot. Well, that's for DNA. Well, we want to look for RNA, so let's call that northern blotting. And then the protein people said, oh, we want to use this blotting technique as well, and we call it western blotting. And so that's how these kind of silly names came to be. For northern blotting, what you do is you make a gel, agarose gel, you load your RNA, you run it through gel electrophoresis, you add a ladder, but at this point you can't see anything, right? Other than the loading dye, you can't see anything. You don't know where your RNA is. And now the gel is set up in a glass dish. This is the glass dish around here. And here's your gel. And you put under it a wick, and the wick is just a piece of paper, of blotting paper. So it hangs into the glass tray, and the glass tray contains a buffer. So the buffer is then wicked through the gel and then onto a membrane. That's the next layer here. So here's the membrane. It's a nitrocellulose membrane usually, sitting on top of the gel, sitting on top of the wick, which hangs into the glass tray. And then on top of the membrane, you put a stack of paper towels, and those are just simple paper towels out of the paper towel dispenser in the lab. You just cut them to size, stick them on top, and that helps now sucking the buffer through the gel, through the membrane, into the stack of paper towels, and by doing so, depositing the RNA from the gel onto the membrane. So it basically sucks the buffer from up from the reservoir, from the glass tray, through the membrane and into the paper towels. And then it deposits the RNA, and then you come back the next morning, you throw away the paper towels, you peel off the membrane, and this is your membrane now, and that contains all the RNA, and you still can't see anything on it. So in order to visualize that, you then put it into a container, and this is a Ziploc bag. This is actually how people used to do it in the past. Then they put it in specially made little rotary or rotisserie glass jars that would go in a rotisserie oven that turns around and sort of lets the probe, the radioactively labeled probe, in a buffer sort of swoosh around the membrane for an hour or maybe a little bit longer. And at that point, all these labeled probes, these little red squiggly lines, are the labeled probes. So that's the RNA, or can be DNA, that's complementary to the RNA that you're looking for, will now find its target, wherever it is on this membrane. And then you can wash the membrane, get rid of all the unincorporated label, and then you have your membrane. You still can't see anything, right? So I'm just putting these red lines on there so that we kind of know where they are, but you can't see them until you visualize them. And then you take the membrane and you put it on a film, you expose that in the dark room, you develop it through a machine, like a photographic machine essentially, and then where the probe bound the RNA, you can see it, and if the signal is bigger, then there was more RNA, and the signal was smaller, then there's less RNA. And the other thing that you can find out from a northern blot is also the size of the piece that you're looking for, because it was electrophoresed through the membrane. And so a larger piece would be further up and a smaller piece would be further down. So that's a northern blot. Now with a western blot, you just, instead of the RNA, use protein. You run it on a different gel. You don't run it on an agarose gel. That's just because the proteins are larger and they're more difficult to separate. So you do that on a polyacrylamide gel. And you've done that, and so you know how to do that. It's also harder to get that from the gel onto the membrane. So you use a machine that uses a much higher voltage. I mean, it uses a huge voltage to suck the proteins onto the membrane. Here, this is just done by wicking. But with proteins, you have to use a lot of voltage to sort of remobilize the proteins. And then with DNA, it looks almost like this. Okay, so that's our northern blotting. And you'll see some northern blots in a second when we talk about these particular aspects of blue light receptor and the work that was done to find out about them. But I said I want to talk about a few RNA quantification methods, and so northern blotting is one of them. Quantitative PCR is another one. That's a little bit more recent that this was developed. So you see in more classic or older papers the northern blotting technique. The quantitative PCR is what's usually done now. It has some advantages. It also has some disadvantages over the blotting technique. But essentially what you do here is you grind up your tissue, you isolate your RNA, and then you do a PCR reaction where you amplify the gene of interest with primers, and you quantify that using a specific machine that can measure exactly how much RNA, how much PCR primer is incorporated with every round of PCR. And the more material you start with, like if you have two samples that you're comparing, the more that you start with, the faster that incorporation will grow. So the signal in that will grow much faster in a PCR reaction, cycling reaction, than one that starts with less material. And so at the end, you can see in that visualization as well which one has more of the two samples that you're comparing, for example. And then a third way to quantify RNA is through something that's called in situ hybridization. We'll see more of that later in the semester, but I wanted to bring it up here because we're talking about RNA quantification methods right now. So why is it called in situ? In situ means in place, but it also uses hybridization. So it's the same general technique as before, where you have the probe that you have to make, but in this case you leave the RNA in place. You leave the RNA in the tissue, you section the tissue rather than grinding it up, and then you have sections, and you can see where exactly in the tissue on the cellular level this particular RNA that you're looking for was expressed. I have a picture for you here to see that. So here's a longitudinal section of a meristem. You see these are the leaves. Here's more leaves, right? And so this is the apical meristem right here. And here are the very tips of the shoot apical meristem. So leaf primordia here, here's probably one, and then here's a leaf primordium, and then these are individual primordia. And the point here is not so much what it is that we're looking for, but the point is that you can look for certain gene expression in the tissue using this technique called in situ. So each of these dots here are the labels that you can see. So in this case, the tissue was sectioned on little slides, and then the slides were incubated with a probe, and then they were washed, and then they were exposed to, it can be actually exposed to an emulsion, a film emulsion, but more often people use an antibody reaction here that simply visualizes the probe so that you can look at it under the microscope. And so these darker dots here is where the probe bound, and all the lighter purple is background binding. So you can see now that where in the tissue this particular gene was expressed. If you had ground up this entire apical meristem, the whole bud or something, you wouldn't know exactly where the tissue was expressing this particular gene. So that's the advantage of in situ hybridization. And then we talked about earlier in the semester about microarrays and RNA-seq, and we used some of that those methods in some of the papers that we discussed. So that's of course also an RNA quantification method. Okay, so enough about RNA quantification methods. There are several, and you should know some of them. You should know all of these really, and be able to determine what the advantages and disadvantages of using each one of these are. Now let's look at some genes using the northern blotting technique to see how specific light quality affects the transcriptional activity of certain genes. So in this example, we have some genes that are expressed under certain light conditions. This is a northern blot gel. So in each of these lanes, an example or an extract of an example was loaded of plants that were grown either in blue light, green light, orange light, red light, white light, meaning it had all the light, or in darkness. And so there are two genes, actually three genes, but two genes that are involved in photosynthesis. It's really not so important in this case exactly how they are involved in photosynthesis. Here we're just interested in the fact that they are specifically responsive to certain light. One's called GSA, the other one's called ALAT. And down here I have the gene names and exactly what they do. The third gene, the G protein here, that is an unregulated so-called housekeeping gene that people put on these gels so that they can show that they loaded equal amounts of RNA onto each of the lanes so that they are comparable from one to another. Right? So if you said, for example, if you said the GSA gene is expressed in green light, but it's not expressed in orange light, then someone might say, well, did you actually load any RNA here in the orange lane? Or did you just forget, or it was just much less, or there was a pipetting error of some sort? And so if you have a second gene in there that is not regulated by light or by anything that you're testing, then you can say, well, but look, I have the same amount of this control, this loading control gene everywhere. So that suggests that I loaded the same amount of RNA overall, and that the difference between this and this lane is in fact real and specific to the light conditions under which the plants were grown. So the point of this slide here is that RNA analysis by northern blotting can be done to determine the expression of specific genes, in this case photosynthetic genes, that are activated by specific light qualities. So in this case by green and blue light, and in this case also mostly by blue and green light, but also a little bit by orange and red, and even in the dark. So in fact, what you could say compared to darkness, these are less expressed in red light than in darkness, but it's expressed more in blue and green light than in darkness. Right? So if darkness is our point that we want to express the other values relatively to, which we could, we don't have to, but if we could say this is less than that, this is more than that. If you compare it to white light, which of course has blue and green light in it, you see about the same response, because that's probably the blue and green light part of the spectrum. And the same is true here, where the white light also contains the blue and green. But you can see that in red light, as well as in darkness, this particular gene is not expressed. Okay, so So we have some genes that are specifically expressed. We have a technique now that we can use in order to determine how the gene expression is regulated. And oftentimes when you see these types of analyses for northern blots, for example, shown in papers, they are actually, sometimes they show you the gels, oftentimes they show you the gels, but sometimes they just show you the results as they get them from densitometer analysis. So densitometer analysis means that you take this gel and you use a computer and you ask it how dense, how many pixels of black are basically found in this section and in this section and in this section. And so then a number is given to you. You can do that with ImageJ actually as well. And then you can graph that number in an XY graph just like here. So for example, in this case, this is a time course for the GSA gene that we had in the previous slide. Previous slide, we had different light conditions. Here we have different time conditions, right? It's all in blue light. At 0 time, the blue light was turned on, and you can see that this light, that this gene is very rapidly induced by light. And at 2 hours, it has its maximum, and then at 4 hours, it's already almost off. At 8 hours, it's completely off. So this tells you something about the time course activity of this particular gene, of one particular gene shown in a densitometer type analysis coming from RNA blots. So just to illustrate that, I put this is an image that I drew, so it's not a real blot, obviously, but just if you wanted to see what the blot would have looked like that gave you this result here, it would have been something like this, right? So if these are the hours, 0, 2, 4, 6, 8 hours, and then this is our loading control, the G protein, then you would see a very dense band, very high pixel intensity at 2 hours. So this corresponds to this. And then very low expression at 0 hours and at 8 hours, and somewhat intermediate down here between 4 and 6. So that's what a densitometer could then do and tell you, and then this is how you would graph it. Or you could graph it in a bar graph, where you can take this, for example, and then say, okay, this is relatively dense, this is relatively dense, so this is these bars, and then low intensity would be these bars, and then and so forth, right? So that's what you could do for relative intensity. And I guess for the darkness here, I forgot to draw the bar on there. Okay, so quick recap so far. I told you at the beginning, plants respond specifically to blue light. For example, while bending, phototropism is one of those responses. And we're trying to find out what the molecular mechanisms of these responses are. And then I said, let's take a step back. We need to understand how to analyze this and how the kind of experiments were done in the past that helped us understand these things. So in order to understand, we first need to understand the mechanisms with which RNA is measured, and I explained to you the northern blots. We talked about qPCR briefly, in situ hybridizations, microarrays, and RNA-seq. And then I said there are specific genes that are upregulated by specific wavelengths, and we used the northern blot as an example to show that, to see how these genes are regulated. Next is the question, what are the receptor proteins for blue light? So for red light, we spent weeks really talking about phytochrome, and you are working with phytochrome in the lab. Phytochrome is the receptor for red light, and there are other receptors in plants that are specific for blue light. So we're going to spend some time today now for the rest of the lecture discussing some of these particular blue light receptors. So if you wanted to find a particular gene for a particular function, how would you go about doing that? What would you do in order to say, okay, I'm looking for a particular phenotype. I know what I'm interested in. I'm interested in phototropism, for example, or something else that's blue light regulated, so that I can identify a gene that is blue light regulating. How would you go about it? Well, we talked about this in the context of finding genes for membrane transporters. So you've seen this slide before. This is the slide that I showed you for the identification of genes for transporters and genes that were involved in salinity, in the salinity response. So you may recall this. This was called a mutant screen. So back then I said you take seeds from a plant, you mutagenize them with a chemical, or you insert some sort of tag DNA that you can later on use. You want to break maybe one DNA on average per seed, but it's always going to be a different one because this is a random process. Then you plant them all out. You screen them under a certain condition, and this is where you have to become creative, right? If you are looking for something that is sensitive to salt, for example, then you would put some salt in there, and you would look in the F2 generation. So after it is segregating for a recessive allele on both chromosomes, for somebody that looks different, right? So in this case, all of them are growing okay, but this guy is looking weakly, and so that must be overly sensitive to the salt that you put in there. And so then you take that plant, you take it out, and you isolate the gene from it, and then you deduce the protein and analyze its function, but you already know that it has something to do with salt sensitivity. Well, in this case, what they did, a very similar setup in general, but they took mutagenized seeds, and the screening conditions here was blue light. So all the seeds were grown, and all the seeds were exposed to this unidirectional blue light. So it only came from one side, and all the plants were growing towards the blue light, with the exception of the odd guys out that weren't doing it. And so those are the ones that you're interested in. So they isolated these particular plants that were not responding by growing towards the blue light, and then isolated the gene of those, analyze the protein function, and that's what I'll show you here in a second. Just want to make sure that you realize that for mutant analysis, just in general, the change in phenotype due to a mutation suggests wild-type gene function, right? So here's the change. That gene that is broken, that does not allow this plant to do what all its buddies are doing, that is the indication of what the gene is that normally allows the wild-type plant to do its job. Okay, so what did that look like when people were doing that kind of screen? So here's an example from Liscombe and Briggs, published in '95. Blue light came from the left side here, and so you see Columbia, COL, that is the name for the wild type that they used. It's a variety, particular type of Arabidopsis seed. So it's called Columbia. That's one that's frequently used. So the wild type here is responding. It's growing towards the blue light, while all these guys here are not growing towards the blue light. And so these are the mutants that they had isolated. Of course, this is not the entire screen. Otherwise, you would see hundreds of plants that all look like the wild type. But you would see the one that is sticking up straight. They took those out and put them next to each other so that you can see the different alleles that they found. And that brings me to a point. I said different alleles. These are all the same genes, right? This is all NPH1. That's the name of the gene. But there are different alleles. Remember what an allele is. An allele is just a variation of the same gene with a polymorphism or mutation, if you will, different nucleotide composition somewhere in the gene. And so the difference between NPH1, NPH1-3, and NPH1-4 is simply that in the same gene, called NPH1, there are 3 different alleles here, so mutations in different parts of the gene. I just wanted to make sure that you recall this type of nomenclature. So what I just told you about is multiple different genes with the same name get numbers added directly. So multiple different genes with the same name, right? So NPH1 is the non-phototropic response. 1 and 2, those would be different genes, 2 different genes. They're just both non-phototropic. But if it's the same gene, as it is in this case here, then multiple alleles will have the same numbers at the beginning, but then they get the dashes added on, and those are the ones that distinguish them. So in this case, NPH1-1, NPH1-3. And this is something I had mentioned before, and I want to make sure that you are good with that you're familiar with that, because it's very easy to be imprecise when you write something. When you mean the mutant and you use a different type of nomenclature, then it's really hard for the reader to understand whether you talk about the wild type or the mutant allele. So just remember that the mutant genes are always in small italics, such as phi A here or NPH1. Wild-type genes are always in caps that are italics. That's the gene, that's the wild-type gene. That's not the wild-type protein. The wild-type protein is in straight caps. And just for photoreceptors, so, well, or any other protein that has a protein bound to another group of some sort, just like photoreceptors often do. Phytochrome A, when you see phytochrome A in straight small letters like this, that means both the protein and the apoprotein and the photoreceptor bound together. Then you write it like this. Okay, so if you write it like this, then you're talking specifically about the gene, not the light receptor part of it. Very important. Do take a look at that and make sure that you understand that, so you can distinguish that when you see it and also when you use it yourself. Okay, so NPH1 is one of those blue light receptors. There were other blue light receptors, and so I'm going to show you a few other blue light receptors. Here's a blue light receptor that's called phototropins. So with phototropins, they found two different types, and they called them both phototropins, and so that's why there's a phototropin 1 and a phototropin 2. And you see here in this slide what its responses are. Wild type is here in this first line, and then phototropin 1, the mutant phototropin 1, you see small italics, is in the second line here, and then phototropin 2. And then they have a double mutant where both of the genes are knocked out together, and this is the response. So there are three different types of phenotypes they looked at: phototropic response, chloroplast movement, and stomatal opening. So let's look at this here. There's weak and there's strong light. And I have weak and strong here as well, and for this is only strong light. But when you have weak light or strong light, the response in the wild type is the same. The plant bends towards the light. But if you have phototropin 1, you see that in weak light, it cannot respond. But in strong light, it responds just fine. And in phototropin 2, in weak light, it responds fine. And in strong light, it also responds fine. So if you just looked at the strong light, you would say, oh, there's really nothing going on, right? This is our response. So turn on the light. I see nothing different from the wild type. So phototropin 1 doesn't seem to have an effect. But when you look at the double mutant here, you see that at that point, the function is knocked out, and there's no longer phototropic response. So that's one kind of response that you can get when you have two genes that are affecting very similar aspects of the same phenotype that you're looking at. Here's another example. So in this case, in weak light, the chloroplasts are arranged either at the top or sort of at the top. But in strong light, at the wild type, for example, you have the chloroplast moving away, and that is, of course, the response to highlight, right? That's a photo inhibition type response where it's trying to avoid that, and it's just moving away the chloroplasts. So that is a response that is mediated by the phototropins. And you can see that when you have phototropin 1, that is still working fine, but if you have phototropin 2, that is not working anymore, right? It's just doing the same as if it was in weak light. So this response of moving the chloroplast out of the way in phototropin 2, mutant of phototropin 2, is not working. But if you take both of them together, then the response looks very different. Then there's not even any arrangement anymore, either at the top or in this slightly differently arranged layer on the top. They're just completely randomly sorted in the cell. And then here's an example where the wild type opens the stomata during strong light. And if you knock out one of the phototropin genes, phototropin 1, you get an intermediate phenotype. It's sort of halfway in between. And the same is true for phototropin 2. And when you knock them both out, then you get complete closure. So this is an intermediate phenotype that you get from each of those two types of phototropin genes. And so, while we're talking about phototropins, I am just using this example to show you some general genetic terms that describe this kind of interaction. So for the stomatal opening, right, for here, you can see that this has what's called an additive effect. So each of these two genes have an additive effect. Both of them together give you the full function of opening, and each of them by themselves only gives you part of it. The phototropism in strong light is an example of clear redundancy. So that means, genetic redundancy means that you have one and that's good enough. If that one's broken, the other one is still there and will provide the function. They're redundant to each other. But if you knock them both out, the function is gone. And then in this example, it becomes a little more complicated. Here you have partial redundancy, right? Because you still get some function. Here you still get some function. You get some function, even though it's the wrong function, but at least you get some sort of an assortment of the chloroplasts. And so you could say there's some subfunctionalization between these two genes. They have different subfunctions. They both act in chloroplast movement, but they have different subfunctions in terms of how they move the chloroplasts out of the way. But they also are redundant in that they move chloroplasts, and if you knock them both out, then chloroplast movement is completely abolished. And so that's, again, an example of redundancy. So I wrote here partially redundant and some subfunctionalization. So you should know what additive gene effects are, what redundant gene effect is between two genes that have similar function and that can be redundant to each other. So in this case, phototropin 1 and phototropin 2 are also called members of a gene family that have redundancy to each other. And they're blue light receptors. All right, third blue light receptor. So in this case, I want to introduce you to yet another photoreceptor, blue light photoreceptor, but I also want to show you another technique that is used here to study these photoreceptors, right? As you will have already noticed, this whole lecture is sort of weaving together some molecular tools with some biological information about light reception. So cryptochromes are yet another gene family of blue light receptors. And what I want to show you here in these two graphs is their function with respect to hypocotyl length and anthocyanin accumulation. So let's first look at the wild type. So if you have the wild type plant, you grow it up, it's a small little plant, grows in darkness, comes to the light at the end of the top of the soil, just breaks through the soil and gets the first rays of light, stops to grow quickly because at this point it has reached the light, it can now produce chlorophyll, it can now make leaves, it can do photosynthesis. It doesn't have to grow as fast anymore as it did when it was underground and had to just use its reserves. So a wild type plant in these blue light conditions will, under the conditions that they were using in this example, grow to a hypocotyl of about this length. This isn't, raw data. This doesn't have error bars or statistics shown on it. What this graph from your textbook is trying to show you is that type of technique where you're looking for a mutant to give you some idea of the function. So you knock out the gene and you see that the hypocotyl is much longer in the cryptochrome 1 gene, right? So relative to the wild type, that plant cannot see the light. It thinks it's still underground. It grows fast and furiously and is much taller than the wild type is. So these are probably relative to each other. This is centimeters, so it's not relative, but you can see the point that they're trying to make, that the mutant is much taller, the wild type is much smaller. And then for another example, for another function of cryptochrome, wild type produces a certain amount of anthocyanin. That's a sunscreen, some sort of a secondary metabolite that allows plants to grow and not be affected by too much light. So plants usually have some sort of anthocyanin. It's a bluish, reddish type of compound. If you have a mutant for the cryptochrome, then it will produce much less of the anthocyanin. So here's a cryptochrome 1 mutant, much less of the anthocyanin. Okay, so the point of this slide is not just to tell you the function of cryptochromes as they are perceiving blue light, but also to show you this technique where people use both a mutant, but then also couple that analysis with something that's called overexpressor analysis. And so in order to understand the function of this blue light receptor, what these researchers, Ahmad and Kashmir, did is they took the gene, so that would be the cryptochrome 1 gene. So we'll call that the native gene X. That would be our wild-type cryptochrome gene. And of course, it has a promoter that's driving its expression, right? So if you have it expressed at its normal level, its native level, then that is what the function is, what the result is with respect to hypocaudal length or with respect to anthocyanin accumulation. So now what you can do for overexpression is you swap the native gene's promoter with a strong constitutive promoter. Constitutive means always on. For example, a viral promoter called 35S. I'm just mentioning this here because that's the one that's most frequently used in plant science for this kind of analysis. But it could be any other very strong promoter. It's a viral promoter. Also doesn't really matter that it comes from a virus, but the virus has this really strong gene that's always on after infection. It just has this particular gene, the 35S gene, always on. But for our purposes here, that promoter, not the gene, was taken. The promoter was taken, and it was fused to the gene of interest. So this is the same gene here. It's our cryptochrome gene, but the promoter has changed. So that promoter is now in overdrive, right? That's a really strong promoter. That's always on. It's constitutive. It will always express this gene. So if you are unregulated in the amount of cryptochrome that you're making, you should show the opposite response of what you are showing when you have none of that protein, right? So if you have the cryptochrome mutation, then you have a longer hypocaudal. If you have a cryptochrome gene in overdrive, you should have a much shorter hypocaudal. And that's exactly what they see. So compared to the wild type, you knock out the mutant, you get a long hypocaudal. You overexpress the gene, you get a shorter hypocaudal. And with respect to anthocyanin, if you have a mutant, you get hardly any anthocyanin. If you overexpress that gene, that cryptochrome gene, you get a ton of that. So in mutant analysis, people try to find the mutant and then they try to analyze the function. And oftentimes, once they have the gene analyzed and the DNA sequence of it known, they can then play with it and they can change the promoter so that they can also study the overexpression phenotype. And that's one of the reasons why I'm showing you this particular example here. Okay, here's one more example of another technique that goes with our blue light receptor analysis that is good for any kind of other analysis as well. But we'll use the blue light as an example. So cryptochrome, we now know what it does, and we know that it is responsive specifically to blue light, but what we don't really know is its mechanism. So how does it function? How does it work? For phytochrome, I told you that there was this change in the apoprotein structure with respect to its photoreceptor, where the photoreceptor had a ring that was flipping depending on the light, the red light versus the far red light. Well, the mechanism here is somewhat different. So what you have here in this particular example is something that's called an in vitro phosphorylation assay. And I'd like you to learn what an in vitro phosphorylation assay is, and we use this as an example for you to do so. So what they did is they took their proteins. So they grow the plants in the dark, and they were growing them on radioactive phosphate. So something had in their nutrient medium had radio-labeled phosphate. So that means all the proteins that incorporate this radio label will become radio-labeled. In other words, all proteins will be radio-labeled. Everything in the plant, after growing them in the dark, doesn't matter that it's dark, but in this case, it does matter because it's a light receptor. So they were growing them, they were feeding them with this radio label. All the proteins will now be radio-labeled, and if you run them on a gel, on a polyacrylamide gel, you would be able to now see all those proteins after exposing that to a film. It's called an autoradiograph, a radioactive photograph, essentially. You would see all of that. You would also see all of the proteins when you stain them with Comasi. So they might be radioactive. You don't see that if you don't visualize that with a Geiger counter or with this autoradiograph. But if you stain it with Comasi, just like you did in cell biology when you ran your Western blots, you would see all those stained gels. You would just sort of see a little bit of everything that's there. And so they did that here, and they show that pretty much as a loading control to say we have about the same protein amount in every lane here. But now they specifically did this experiment with wild type in darkness. So they kept these plants that were growing in the dark, or they exposed them to light. And they did that with the double mutant Cry1, Cry2 in the dark or the light. So when you take your plants, you grow them in the dark, you feed them with the radioactive label, and then you put them in the light, there's one protein that seems to be picking up a ton of this P32, this phosphorylation label. And so one particular protein is now super phosphorylated, right? Picks up all this 32P. Seems like there is some sort of phosphorylation event going on for this particular protein. Well, when you do that for the mutant, that phosphorylation event does not occur. And so you do not get the phosphorylation because you are missing these two proteins. So if you now extract the total protein, you separate the protein mix on the acrylamide gel and you expose that gel to the X-ray film, you have this analysis, you can tell that this must be the Cry1, Cry2 protein because that's the only thing that's different in these two assays. And the other only thing that's different between these two lanes is whether or not they were exposed to light or not. And so in the wild type, this particular protein becomes phosphorylated. That particular protein, that lane here, that place here is not active. It's not functional. The protein may be there, but it's not functional. It does not autophosphorylate. You do not see the blob here. Right? So this suggests that the cryptochrome gene works by phosphorylation, by autophosphorylation with respect to light, and then as a phosphorylating cascade, it phosphorylates the next signal transduction member downstream. But that's not part of the discussion right here. So what I showed you is that there are three different types, three different families of blue light receptors that are all active with respect to blue light specifically. I showed you some ways with which you can determine gene expression, northern blots and other types of RNA analysis. I also showed you some ways how you can find the mutants that are exposing the gene function that you might be interested in this case blue light. And I showed you some ways how you can describe the different types of genes, whether you have redundancy in a gene family with respect to a particular function, in our case for blue light, or any other, of course. And then I showed you some ways how you can analyze a gene once you have it, once you have the molecular identity determined. So you can, for example, do an overexpression analysis, or you can see whether or not this gene is phosphorylated using an autophosphorylation assay or a different type of phosphorylation assay. But in this case, nothing else happens to it other than it gets activated by blue light, and that alone is enough for phosphorylation. So autophosphorylation occurs. Okay, so I want to show you one last thing, and that is an example of an important function that is specific to blue light and that tells us, puts a few of the things together that I discussed with you in this lecture. This comes from your book. You can go back and read all the details about it, but what it explains is how blue light is acting in such signal transduction pathway, for which I just showed you bits and pieces here, in entire signal transduction pathways with respect to blue light perception and then regulation of the stomatal opening, which of course is really important so that the plant can regulate how much water it picks up or evaporates while it's picking up carbon dioxide. So regulation of stomatal opening, of course, is very important so that the plant doesn't die from drought stress. So here you have the phototropins. So that's the phototropins genes, the proteins that I discussed with you earlier. Phototropins absorb blue light and they also autophosphorylate. I showed you an example for the cryptochromes, but we could have done the same experiment for the phototropins. So they autophosphorylate, and at that point they activate a downstream molecule. This is called BLUS1. Not so important what this is. Phosphorylation, activation of this particular protein. This protein now signals to another regulator, and this regulator has two particular parts, not so important what they're called, but this regulator, this intermediate signal transduction molecule, now activates another kinase. This is not even known, but it is known that these two don't interact directly with each other, therefore something has to be in between. But the point is that this phosphorylation event here, through this cascade of events, then triggers this HTPase activity where hydrogen is pumped out of the cell, and that causes, together with this other protein here, the hyperpolarization, or the difference in charges on the outside versus the inside, because all these hydrogens are pumped out. So all of these proteins are pumped out. And when you have a hyperpolarization, if you have tons of positive charges up here and negative charges inside, that drives anything that's positive out here to inside the cell to even out that charge differential. And that, of course, results in the decrease of the water potential inside here. And that leads to uptake of water then into the cell. And as the cell swells, it exposes the stomatal opening between these two guard cells. That's how opening of the stomatal pore works. There's another important part, and that is the use of this little hormone here. It's called abscisic acid. We'll come back to that later, but abscisic acid interferes with this particular pathway here that is blue light regulated. So blue light opens up the stomata, but ABA, which is produced under drought conditions, will do the opposite. It will inactivate this intermediate so that it interrupts this signaling pathway, so that water no longer moves into this cell and opens the stoma. But now this is interrupted. No more water moves in, so therefore the stomatal opening does not open. And in fact, the opposite happens. It activates this anion channel where exactly the opposite occurs, right? So here's a positive charge that's coming out that's causing the water to move in. Here's negative charge moving out, changing the pathway in the opposite direction. And so ABA will activate this, phototropin will deactivate that. So all of these together with this hormone, together with phototropin, with the blue light reception and all these intermediate pathway steps, and then the membrane transporters and the water potential changes, all of these things that we have discussed so far, will then regulate the opening of the stomata under certain environmental conditions, light and drought. Okay, so I just put together a few things here from what I just told you that I thought was important. You can read this up here, and you can also go to your book and look at it that way. But what I wanted to make sure that you get from this lecture is a number of things. One of them is that there's blue light receptors, there's specific responses to blue light. One of them has to do with sensing the environment, water and the stomatal closure. And it puts together a lot of different aspects of plant physiology that we have discussed so far. The other important part of this lecture was that I wanted to use it as an example to talk to you about a bunch of different techniques. And all of these techniques are important to know because that is how we know what we know, right? And it's not really enough to just know stuff. What I really would really like you to learn in this course is also how we know the things that we know, so that you can be in the future confident to just pick up papers and look at the techniques and learn stuff for yourself without having to memorize the outcomes of any of these things that you could look up easily by yourself. So that is why I do want you to remember the types of RNA experiments, why phosphorylation experiments are important, why overexpression of certain genes as a way to understand function is just as important as mutant screens and how mutant screen works. I'd like you to know about the genetic interactions of redundancy and gene family members that have slightly different functions but can subfunctionalize or have completely redundant function. So all of that together was built into this lecture of blue light. Okay, That is it for today. I'll see you in class.